In this video, we will study what is secondary growth, how it occurs in dicotyledonous stem or dicot stem. If we see dicot plants, mango, neem, pulses, gram seeds, kabuli chana, black chana, kidney beans, sunflower, there are many plants which are dicot plants. Now, what are dicot plants? Dicot plants are plants which have two cotyledons. Now, we can see here example of gram seed. This is gram seed. Now, when we soak the gram seed, open it up, we will find two cotyledon, yellow in color, and in the center is present embryo. This is embryo. Like we can see here also in this figure, this is the seed and these are two cotyledon, and in the center is present embryo. So this is called as dicot seed. To study secondary growth, we'll take sunflower stem again. Now the sunflower stem here, if we cut its section, this may show primary structure, this may show secondary growth. Now what is primary structure and what is secondary growth? This we'll study in this video. Now if we germinate the seed, right? So first thing which comes out during germination is radicle. Then plumule comes out. Radicle forms root system and plumule form shoot system. So this is the shoot system and this is the root system. So what is shoot system? This is stem, branches, buds, axillary bud and leaves. And this is the root system. Root system is taproot system. Now this plant which is growing, this stem and root is showing primary growth. So this is the primary growth. I mean, this is first time growing, first time developing all the tissues, xylem and phloem. So this is primary growth. Later on, when this plant will grow, this will also increase in height as well as its diameter will increase. So this longitudinal growth which is occurring, this is called as primary growth. But when its diameter start increasing, branches as well as main stem, then it is called as secondary growth. So here we can see diameter is increasing, length is increasing, root system is also being broadened up. So we can see this plant may turn into, if we talk about mango, that is also a dicot plant, this may turn into a tree. And if we see the tree, it has a stem or trunk which is more in diameter, and this is its canopy, broadened canopy, which is again supported by branches which are more in diameter. So this is to support the tree growth. So this is called as, this increase in diameter, formation of wooden branches and supporting the canopy structure, this is called as secondary growth. So first growth which occurs, that is called as primary growth, and later on which growth occurs and increases the diameter of the plant, this is called as secondary growth. During secondary growth, secondary tissues will be formed, secondary vascular tissue, secondary parenchymatous tissue, secondary xylem, secondary phloem, these will be produced. So we can see here in this figure, this is the primary structure and this is the start of the formation of the secondary structure. Ultimately, if this is mango or some tree growth, this may develop into a tree. So this is called as secondary growth. So secondary growth will lead to increase in the diameter. This is called as isodiametric growth. And ultimately, this will also helps in wood formation. What is wood formation? Basically, this is formation of secondary xylem. Secondary xylem, first formed xylem is called as primary xylem. Later on formed xylem is called as secondary xylem. Now, if we cut the section of the stem showing secondary growth, we can study the mechanism. How does this occur? First, we'll take the stems which are showing primary growth. Then we'll take the stems which are showing secondary growth, and we can understand the mechanism how secondary growth occurs. Now in this slide, we'll study how it occurs. Now if we cut the section of the primary stem, we will see that when section is cut, this is called as transverse section. And we will see that during transverse section, all the vascular bundles, they are arranged in a ring. To know more about the primary structure of the dicot stem, you can watch my video of anatomy of dicot stem. Link is shared in the description box. We can see here vascular bundles, they are arranged in the form of a ring. Now if we see each vascular bundle, this has cambium in the center, and this part is xylem, this part is phloem, this is its pericycle, and this is the endodermis, which is delimiting the vascular tissue from the cortical zone. This cambium is very important for the secondary growth. This cambium is also called as intrafascicular cambium. Now this arrangement of xylem is end arc because metaxylem is toward the outer side, protoxylem is toward the inner side, and this is the xylem parenchyma. So this phloem, this is primary phloem, this xylem, this is primary xylem, and this is the cambium which will help in the secondary growth. Now this is the structure of the primary Dicot stems vascular bundle. Now we can see here all the vascular bundle, they are arranged in the form of a ring, and each vascular bundle has a cambium which is called as intrafascicular cambium. Now what happens during secondary growth? When secondary growth occurs, now this zone, which is parenchymatous in nature, now this will again turn meristematic in nature, and we will get a complete ring of the vascular cambium. So this zone is called as intrafascicular cambium. Now this will be called as, this part is called as interfascicular cambium. Interfascicular cambium, which is being produced between the two bundles, like here, this will be produced like this. So ultimately, this will be converted into this type, complete ring of vascular cambium. Now this complete ring of vascular cambium, this is also called as vascular cambium, which is formed by intrafascicular cambium and interfascicular cambium. Now if we see the structure of the cambium, this is just meristematic in nature. This is like this. All cells, they are parenchymatous in nature, having cytoplasm and nucleus. Now this, if we take one cambial cell here, supposing if we take this cambial cell, so this is like this, this will be having nucleus. Now we can make nucleus over here. Okay. Now this cambial cell will cut off, this cambial cell will divide like this. If it is divide by like this is called as periclinal division. Periclinal means parallel to the wall, and this will cut off two cells, right? Now this, when this is cutting off two cells, now this internal cell, this is called as xylem mother cell. Why mother cell? Because this will lead to the formation of the xylem tissue, and this cuts off again, one periclinal division will occur, and this will cut off phloem mother cell. Now this is the phloem mother cell. Phloem mother cell because this will form whole of the phloem tissue. So this is the cambial cell which is cutting off xylem mother cell toward the inner side and phloem mother cell toward the outer side. So cambial cell is meristematic in nature. So this is the vascular cambium which is basically cutting off secondary xylem toward the inner side and secondary phloem toward the outer side. Now we can see here when we again cut transverse section of the stem. Now this is TS of stem, Dicot stem, showing secondary growth. Now this is showing secondary growth. Now we can see here, this is the vascular cambium which is present in the center. Now this was cutting off secondary phloem toward the outer side and secondary xylem toward the inner side. So we are getting a ring of secondary xylem and a ring of secondary phloem. Primary phloem is being pushed toward the outer side, and this may get converted into fibers like this. And this is the primary xylem which is present toward the pith in the center. And this is the pith part, and this is the cortical zone of the stem. Now when diameter is increasing, because this is leading to the increase in the diameter of the stem, when there is increase in the diameter, now this zone which is called as cortex zone, here again a cambium develops. This is called as cork cambium. Now this cork cambium, this is also meristematic in nature. This cork cambium is also meristematic in nature, which is developing into the cortical zone. This is the cork cambium. So in this way we can see here primary xylem is pushed in the center. This is a formation of secondary xylem, formation of secondary phloem, and primary phloem is being pushed, and this primary phloem cells may get converted into fibers. So this part is epidermis, this is cork cambium, this is pith region, this is vascular cambium, secondary phloem, secondary xylem, primary xylem, and this is the primary phloem, and this is the cork cambium. Now again diameter will increase, and what will happen? We can see here. This is the diagrammatic sketch actually. This cork cambium will cut off cork toward the outer side and secondary cortex toward the inner side. Now this is the cork tissue which is being cut. Now in certain places we can see loosely arranged cells called as lenticels. We'll just study the cellular details of this diagram and we will understand all the things. Now this part is called as cork, and this is interrupted at places by means of lenticels which help in gaseous exchange. This is the cork cambium which will cut off secondary cortex toward the inner side and cork cells toward the outer side. So this is how secondary growth occurs, and we can see here diameter of the stem has been increased. This diametric growth is to meet the demand for increasing growth, for more vascular tissue. So that's why new vascular tissue is being produced every year, new secondary xylem and new secondary phloem. So this is the diagrammatic sketch. If we take a part of this and we see its cellular detail, we can see here. This is the cork cambium. This will cut off secondary cortex toward the inner side, and this is the cork cells which is being cut toward the outer side. Epidermis is ruptured, and at places we can see here loosely arranged cells having intercellular spaces. These are called as lenticels. They help in gaseous exchange, and this part is also called as periderm. This is also called as periderm. So we can say, what is periderm? Periderm is made up of cork cambium. Cork cambium is called as phallogen. Cork tissue is called as phallum, and secondary cortex is called as phalloderm. So this whole tissue which is present at the periphery, this is called as periderm. Now we can see here, this is the cortex which is primary in nature, primary stem cortex. So this is parenchymatous in nature, helping in storage of the food material. Now this is the sclerenchymatous pericycle of the vascular tissue, and this part is called as vascular cambium, which is cutting off secondary phloem toward the outer side, and this is the secondary xylem toward the inner side, and this is the crushed primary phloem which is being crushed and getting converted into fibers. Now this secondary xylem which is being cut toward the inner side, its growth also varies. We can see here, we can see some of the vessels, they are short in diameter. This is during the autumn period. Autumn period is when all leaves fall down, there is less supply of water and minerals to the plant, and they will develop less diametric growth. And when they are entering into the spring season, that is growth season, vessels diameter will increase. We can see here, this is the more diameter of the vessels. So this wood or this secondary xylem is produced in the autumn season, and this is being produced in the spring season. So in 1 year, this is the autumn secondary xylem, this is the spring secondary xylem. So this is also called as annual ring. So this part is primary xylem, which is being pushed toward the inner side, right? And this is the first year annual ring. In the first year, annual ring is produced. So what is annual ring? Autumn secondary xylem and spring secondary xylem. And in the second year, in the next year, again, cambium will cut off secondary xylem and secondary phloem, and old primary phloem or old secondary phloem will be crushed and new secondary phloem will be produced. Now this is the second year annual ring. Again, this is being produced in the spring season, and this is being produced in the autumn season, and in the center is present pith. So this is the structure of the TS of dicot stem showing secondary growth, and these are its cellular details. When we show the structure in the cell formations, this is called as cellular details. Now if we compare both the structures, primary as well as secondary, we can see here. Now this is the primary structure, this is the epidermis, cortex, this is primary cortex, endodermis, sclerenchymatous pericycle, primary phloem, primary xylem, and this is the pith region. and this part is the parenchymatous in nature. Now, if we want to study in detail TS Dicot Stem primary structure, we can again watch my video. Link is shared in the description box. Now, if we compare both of them, right, we can see here this part, sclerenchymatous pericycle, is as such. Now, this primary phloem, this is being crushed, and this is and will be converted into fibers, and new secondary phloem is being produced, and this is the cambium which is helping in the secondary growth, and this is the xylem which is being pushed toward the center, and new secondary xylem is being produced with the help of this cambium, and this part is pith which is here as such. This is the pith, and diameter, supposing if initially diameter was this, supposing initially diameter of the stem is this, and this will increase up to this, or this may increase more, right? So this is increase in the diameter of the stem. So this is also called as isodiametric growth or increase in the diameter. So to meet the supply demand of the increasing tissues, more vascular tissue is being produced every year, which is called as annual ring. So we can also count the age of the plant by counting the annual ring, because every year new secondary growth is being produced, new secondary vascular tissue, new secondary phloem. So these are called as annual rings. By counting the annual rings, we can count the age of the plant, and this study is also called as dendrochronology, when we can count the age. This is called as dendrochronology. Now again, we can see here what is the significance of this secondary growth. Now, when primary growth take place, requirement is less, but when secondary growth take place, requirement is more. So what happens? Older vascular tissue that will become non-functional, and new vascular tissue is formed every year to meet the demand supply. This helps in meeting increased demand for long distance transport of water, minerals and nutrients. And new vascular tissue leads to the isodiametric growth. This will increase the diameter of the plant. This also helps in providing support to the new aerial branches. Like supposing if we have this tree, and this is, if we have this tree, I'll make it again for you. Right, this is its canopy. So to support this stem and to support the branches, right, so we will have secondary growth or pillar branches we can have. So this helps in providing support to the aerial branches. Cork cambium, because this part, if we see the stem, now this is the stem, and its outermost covering will be made up of bark tissue. This is bark which is present toward the outer side. Basically this is produced with the help of cork cambium. This helps in formation of bark which is present toward the outer side, and this will protect the internal tissues from the extremes of environment, as well as from the pathogens. They may not infect the plant, as well as from the microorganisms. So when plant is growing straight, this is called as primary growth, but when its diameter is increasing, this is called as secondary growth, and that is also to sustain the plant in the environmental extremes of the environmental condition, as well as to meet the increasing demand of the nutrients as well as water and minerals. So this is all about secondary growth in the dicot stem. If you like my video, please like and share and subscribe. So thank you for watching my video. If you have any questions, you can ask me in the comment box.